Hi everyone, welcome to Lecture 3, Data Standards for Transcriptomics in Network Biology. In the last lecture, we looked at different transcriptomics technologies, spotted microarray, Affymatrix, and also the latest technology, RNA-seq. We looked at sort of pre-processing aspects, why normalization was important, why biological replicates are important. And in the end, you end up with a gene expression matrix, giving you information about each sample in terms of the levels, the expression levels, usually given in log2 expression levels of all of the genes in that sample. Now, it's quite common for machine learning just to take that matrix of numbers and start doing machine learning on it. So the GOLUB data has been used extensively for testing machine learning approaches, and it is just the gene expression values as a matrix with labels for the samples. I want to, in this lecture, show that there's actually quite a lot more to biological data making up microarray experiments than just that array of numbers, and that actually including some of this, well, some of the data is important to include because it actually tells you about what samples are and how they are different in terms of different factors, but some of it's optional, telling you about differences in terms of genes. Some may be oncogenes, some may be tumor repressors, some may be metabolic genes. And I would argue that actually including a lot of this prior information generally would improve what you can do in terms of machine learning with omics data. Okay, so we'll start talking about data standards. So often information about microarrays, certainly in the past, was acquired by looking at the original research papers. So this is the original research paper by Golub et al. The lead researcher was Eric Lander. Molecular classifications of cancer, class discovery and class prediction by gene expression monitoring. It was published in Science, which is a leading journal, in 1999. Now, at that time, there were no microarray standards, and a lot of microarray papers basically presented information about the studies as they felt fit. There was no consistency, and generally you would need to read the whole paper to actually work out what the data samples represented. So in this case, we have some of the relevant methods here. Our initial leukemia dataset consists of 38 bone marrow samples, 27 ALL, 11 AML, obtained from acute leukemia patients at the time of diagnosis. RNA was prepared from bone marrow mononuclear cells, was hybridized to high-density oligonucleotide microarrays produced by Affymatrix and containing probes for 6,817 human genes. So at that time, 1999, Affymatrix had been going for a while. It was started in around 1992, but clearly the human Affymatrix chip wasn't so extensive. It only covered around 7,000 human genes. Today's chips cover all 25,000 human genes. But this is what you would need to find in the paper, that there's 27 ALL, 11 AML, and are there anything important factors about those samples? Some of the things which affect gene expression, well, gender would be one. sort of male-female patients will have different gene expression profiles, particularly with the Y chromosome being expressed only in males. So there are important factors that can be gleaned off the paper. Even today, going back to the original research paper is often required to actually work out the exact details of a study. The whole idea of developing data standards was to make omics data transferable between experiments and to also make it transferable between data processing techniques. So that in the Golub's time, 1999, you basically read the paper to find out what the experiment was about. But there was limitations to that which was recognised. One of them was that the papers had no consistency. They had no consistency about what information they gave you about this study, and some papers just lacked key vital information which made the data unusable in some ways for downstream analysis. Another issue was if you wanted to do a classifier, a universal cancer classifier. If you took this approach, you would have to read all of the papers about all of the cancer data sets that you wanted to use and somehow synthesize the information from all of them. That would be a massive undertaking if there was about 50 different cancer data sets that you actually wanted to use. So it made what are called pan-cancer studies very difficult, studies that went across different cancer types. So the question, which you can't really see because I'm still here, I should disappear in the next slide, is what makes transcriptomics data difficult to handle compared to sequence data? And that's what we'll look at in the next slide. So if we look at a comparison between genomic data and microarray data, as you would have seen from David's lectures, genomic sequence data is just a sequence of nucleotides A, T, G and C, adenine, thymine, guanine, cytosine. So it's just a sequence like ATG, AT, CG, etc. Although there are ambiguous codes to represent any purine, any pyrimidine, or any nucleotide. But it is very well defined, it's just a sequence. As I'm hoping the lecture about microarrays and transcriptomics revealed, the raw data for microarrays is not so well defined. Spotted microarrays give you the gene expression of one sample compared to another sample. So you can say that a certain gene, like actin, is twice the expression in one sample compared to another sample. But there is no absolute measure of how many mRNAs are within each cell in a sample. So it's all relative to within the experiment to possibly a common reference, but there is no universal sort of comparison between experiments. The data is also very noisy, floating point numbers where each gene potentially has different levels of noise. So that's one of the difficulties. The other key difference is that sequence data really only depends largely on the species sequenced. So we have the human genome, the mouse genome, the yeast genome. So it's largely just the species. There are small variations between individuals within the species. So you might be aware that there's databases for SNPs, single nucleotide polymorphisms. So essentially mutations which make us all different, different hair colour, different eye colour, and there are other differences between our genomes and between the genomes of different species or the same species. So different strains of mice will have different genomes, slightly different genomes. But in terms of microarray data, really the data depends critically on all aspects of the context of the experimental conditions. So not only the species of the tissue which is being microarrayed, but the cell types within the tissue, the actual type of tissue. Is it muscle? Is it liver? Is it brain? The treatments which were applied, how it was extracted, the experimental design. Essentially, all of this contextual information needs to be recorded for high throughput omics data so that we can do sort of sensible experiments with the omics data, a sensible data analysis. So that can be difficult. And then the third aspect is how complex the annotation of the data is. So raw sequence data is generally the key thing being analysed. So you might do gene detection to detect the genes. You might then do translate those nucleotide sequences for those genes into protein sequences, and then maybe secondary structure prediction of the protein sequences. You end up with annotation. So the gene detection, you annotate a gene's location with start and end nucleotide positions. The secondary structure, you annotate the protein sequence annotation, the alpha helix positions, the beta sheets, the coil. But it's not so complex in terms of the actual semantics of the data. Whereas raw data in microarray experiments really depends on a lot of the contextual information of the experiment, as I said up here. So the semantic content of this information is extremely rich and diverse. Essentially, for microarray experiments, you need to be able to describe any molecular biology experiment, because you can do any biological experiment and then do a microarray of the samples resulting. So it's incredibly diverse and rich, sort of the metadata for a microarray. This is really what this lecture is going to be about. So, experimental design, what does that mean? It's essentially how different biological extracts are put onto, say, a microarray, and then hybridization is carried out, and then the resulting data and how it is processed. So in this experimental design, we've taken sort of Homo sapiens, so human liver 1, liver 2 samples. We've extracted out the messenger RNA, converted it to cDNA, using some protocol which may be different between experiments. This then indicates that this extract and this extract were both joined together on one single array hybridization, and they were both dyed with green CY5. So this is essentially pooling. You're actually pooling two samples of liver and you're putting it onto one microarray. And the red channel is an extract reference. So this is labeling with CY5. And then another part of the experiment, you've taken kidney sample 1, kidney sample 2, extracted out the mRNA again, put the two pooled samples together, dyeing them green, and again compared it to a reference. And then the last part, two brain samples, extracted the messenger RNA, pulled the two together with a green dye, and then used the reference again. So that results in 3 hybridizations using a particular array design, SMD10K, resulting in 3 text files containing the log ratios of all of the genes which are on these chips. And this is your final output. So actually to know how to analyze this final output, you really need to know the design of the experiment, that you're actually taking 2 different liver samples and you're joining them together to compare against a reference. That actually changes the statistics that you need to use to analyze the data. Here's a different design. So both of these were extracted from Array Express, which is the database for microarray experiments. So in this case, we've got some undefined source 1b, sample 1b, extract 1b, and we've got source 1a, sample 1a, extract 1a. And this sample is labelled both with red dye and green dye. And then this sample is labelled by green dye, which is compared to the red one on hybridization 1. And this is then labelled also red, which is compared with the green one. So this is actually a dye swap experiment. You're comparing extract 1A with 1B using red on this one and green on this one, and green on this one and red on this one. Again, you've got then a source 2A and 2B, you're doing the same, and you're also doing a dye swap, resulting in another 2 hybridizations. So you get data out of all of those, and again, you need to know the experimental design to know how to analyze that downstream data. So all of this complex data representation was initially tackled by this society which grew up around 1999, at the same time as the Golub paper was being produced. And it was originally called the MGED Society, the Microarray Gene Expression Data Society, but they actually broadened their horizons to also include sequence-based data, sort of next generation sequencing with standards such as MinSec-E. So they eventually renamed themselves FGED, the Functional Genomics Data Society. So they were vital for coming up with a number of standards which made the genomics data, microarray data initially and then functional genomics data later, sort of open and reusable by the community. So they produced open standards. And we'll talk about MIAME, MAJ, MGED ontology, and also MAJ-TAB as some of the key standards that they produced. One of the first standards was MIAME. So this stands for Minimum Information About a Microarray Experiment. And this was quite novel at the time, that you actually need to define what information needs to be stored about any particular experiment. Up to then, we had lots of different papers about microarrays where they were giving different information. Some were missing out some vital information to make it repeatable or to make it easier to interpret, and others were including other information, and nobody really knew what information needed to be put into the metadata of the gene expression data to make it useful for the community and to be able to repeat experiments. And so they came up with this idea of a minimum information, so a minimum standard of what needs to be definitely included. So we have the gene expression matrix containing the gene expression data, but what else do we need to know? So there are two clear dimensions. There's the genes here, and for each gene we want to know the gene annotation. So there's lots of annotation associated with any particular gene within an organism. And then we've got the samples, and for each sample we need to know the sample annotation. So again, there's lots of information about samples. So what data do we want to represent? So the MAME standard splits up the data into these 6 broad categories. The first one is experiment. This provides a description of how the experiment was actually run in the context of the experiment. This could refer, for instance, to a publication describing the experiment. Again, we're on to natural language, but it also allows more controlled descriptions of the experiment. For instance, the Miami standard suggests, when using controlled vocabulary, that it takes a qualifier value source triplet. So you have a qualifier which tells you what the thing is, like cell type, and then you have the value, epithelial, which is a type of cell, and then you have the source of the annotation. So in this case it's actually a book, Gray's Anatomy 38th edition. Similarly, the qualifier might be a treatment that you apply to a sample. The value might be 15 minutes heat shock, which is a type of treatment, and the source might actually be a publication, for instance this one from Nature Genetics. So the experiment allows a more controlled vocabulary description. What does the experiment consist of? Well, normally you take a number of samples, and each sample needs descriptions. In this case, an example description might be the source, NCBI taxonomy. So that actually describes the organism of the sample, the biological organism. So the NCBI taxonomy, it could be Homo sapiens, so it's samples from humans, or it might be mouse or yeast. So the experiment might consist of mixed samples, as in some samples are human, some are mouse. So that's the samples. And then the samples are applied to arrays, microarrays. So there has to be a description of the array, and this is usually what genes are spotted where on the microarray. And for each gene, annotation describing what was the sequence, for instance by giving a GenBank identifier. I think David has introduced you to GenBank. What is the experimental design? Well, it describes how the samples were dyed and then which samples went onto which arrays. So this is really described by the hybridization. What samples were hybridized onto which arrays? So if it was an Affymatrix array, there would just be one sample hybridized onto it. If it's a spotted microarray, it would be 2 samples. And then the experimental design, is it pairwise comparison? Is it a dye swap? Is it a comparison between a reference sample? So there's lots of detail about what hybridizations were actually carried out. Once they're carried out and the microarrays are scanned and the spots are sort of segmented out and the intensities are integrated to get quantified values of the intensity of each spot, then we have to, as we've described in the previous lecture, we have to worry about normalization. So this is the detail of the processing which was applied to the raw microarray data to get final microarray data. The normalization, it might be medium normalization as we've discussed, or it might be low S normalization. And this finally results in data, which is your gene expression values, the matrix of gene expression values, but is now given with all the sample annotation, with the array information, with experimental details, with the experimental design, what hybridizations were carried out, and what normalization was carried out. So there's quite a lot of detail specified in the minimum information about a microarray experiment standard. Once they had come up with the MIAMI, the minimum standard for what data needed to be represented, they then moved on to how do you represent the actual data itself. So this became the MAGOM standard, Microarray Gene Expression Object Model. So they took a rather unusual approach for sort of bioinformaticians, the bioscience community, that they used unified modeling language, which some of you may be more familiar with as sort of software engineering of a class object-based modeling. But they used it to represent the microarray experiments. So they had relationships such as biomaterial and their inheritance, relationships with biosource, biosample, labeled extract, are all types of biomaterial. The relationship here is that biosource is something which is it's a source of biological material, and once you've got a sample of that, it becomes a biosample, and once it's labelled by, say, red or green dye, it becomes a labelled extract. So these are all sort of stages in the experimental process. To actually go from, say, the biosample to a labelled extract, you have to apply a number of treatments to the biomaterial. So they've got the concept of a treatment which involves applying compounds to the biomaterial, such as the dyes. And then a lot of these can have descriptions attached to them, either involving database entries or ontology entries, which we will discuss, or there may be bibliographic references. A paper might actually describe what the treatment was or involved. So this is just 2 diagrams, but the MAGOM model is incredibly complex. It involves 132 classes in 17 packages. So it's just a vast UML class, static class diagram. This was to try and include all possible biological experiments in terms of a description with UML. One of the issues is that it just was potentially too complex for bioscience and also bioinformaticians to fully understand. how do we interchange the data? So the whole idea of coming up with these standards is to allow experiments to interchange data, that we have open standards. So one experiment will produce data in a standard format that can be used by other experimenters. So they also came up with MAGML, the microarray gene expression markup language. And again, they used quite modern technologies at that time. They used XML metadata interchange language, XMI, to describe the MAGOM UML model. By doing that, they could then process this XMI file using the standard object modeling group software and turn it into a representation of the data model as XML language, given as a document type definition. So this shows a small sample of one package. So this is a biomaterial package, and its biosource is identified as human breast tumour, and it's got some characteristics associated with it. These are given as ontology entry. Species is Homo sapien, tissue is breast, disease state is tumour. And then there's other other descriptions. So that's the biosource. The biosample is from is human breast tumour, and it's got an audit trail. So it was created in 2001 by Helen Parkinson, who's an annotator that works at the EBI, and it's got material type, tissue sample. So it's a tissue sample which is a biopsy. So this just carries on. It gives a full description of the biomaterial that was used in a particular experiment. Now, I think it became, so I think this is a sort of lesson in terms of over-engineering. It became clear that the MAGML standards, even though they were very inclusive and could potentially describe any molecular biology experiment in detail, they were just too complex for labs to adopt. That the bottleneck was very much trying to get bioscience experimenters, researchers, to use these standards. So eventually they developed something called MAGTab standard, which then was extensively used. It was basically a tabular format. So that previous experiment with all of this, with liver samples, kidney, brain, would be represented in MAGTab as, well, sample ID, liver 1, Homo sapiens liver, what protocol to use to extract it, what protocol to use to label it, which hybridization ID did it go on. This was hybridization 1, the array design and the output file. And then what else got put onto hybridization 1? Well, liver 2, Homo sapiens liver, these protocols, CY3 hybridization 1. What else was on the hybridization 1? Well, the extract reference. Using this protocol to dye it with CY5, it went on to hybridization 1 to produce the output file 1.txt. And you might see this is incredibly inefficient for in terms of the redundancy. So if you're repeating Homo sapiens on all of the samples, you're repeating the protocol on all of them. It's just not very efficient, but it's something that bioscience researchers that use Excel a lot can actually work with. And it actually contains, or they made it contain, all of the information that was actually the large quantity of the important information that was in the UML standards. So these can be huge tabular spreadsheets, but it's just a simpler way for bioscience researchers. So in a sense, the lesson here is that simpler is sometimes better when it comes to data formats, even if a database engineer would cringe at this type of sort of format for data representation. So a fourth standard that MGED Society worked on was called the MGED ontology. For successful interchange of data, we need to adopt standard vocabularies, and this is what this provides. You may have noticed on the MAJML that we have these ontology entries: species, Homo sapiens, tissue, breast, disease state, tumour, tissue sample, biopsy. So these are the MGED ontology terminologies being used. So you may notice that even simple things like disease state, tumour, the tumour is spelled with American spelling, because that's the sort of common usage is for scientific writing to use American spelling. But even that, if that was used the British spelling, it can lead to confusion for data interchange. So the MGED ontology standardizes the terminology being used. So the Stanford Knowledge Systems Lab was one of the first labs that were looking at ontologies and representing knowledge with them. So they came up with this: an ontology is an explicit specification of some topic. For our purposes, it is formal and declarative representation which includes vocabulary or names for referring to the terms in the subject area, and logical statements that describe what the terms are, how they are related to each other, and how they can or cannot be related to each other. Ontologies therefore provide a vocabulary for describing and communicating knowledge about some topic and a set of relationships that hold among the terms in that vocabulary. And usually they seem to always focus on wine as an example, but you can see here this is an example ontology about wine, and we've got classes, which are the black boxes. So winery is a class, Bordeaux is a class, Pollack is a class, and we've got instances of those classes, red boxes. So Chateau Lafite Rothschild is an instant of a winery, and Chateau Lafite Rothschild Pollack is an instance of a Pollack. We've got attribute slot properties, for instance produces, so this winery produces this wine, and then relationships is a kind of, so these are relationships, facets. So there's a formal way of representing the terminology that will be used in a particular area of knowledge. And that's what the MGED ontology group did. They developed an ontology for describing, or for use in describing microarray experiments. I thought it would be worthwhile finishing this section with a description of the key microarray databases. One of them is ArrayExpress, which was developed at the EBI by someone called Dr. Alvis Brasma, and he was also responsible for a lot of the MJED standards. So he based this database on the MAJOM standard and used it to, or the XMI file, to automatically generate a SQL database schema from the MAJOM standard, and also an object model, so that there was an object relational data conversion. So you could store MAJOM data as MAJML markup language into this SQL database and take it out in terms of the object model. So there was this flow of data. One of the aspects here, though, that we know is the MAJOM standard is incredibly complex with over 100 classes. So actually, bioscience researchers found it quite difficult to describe the experiments, and there were not so many experiments being put into this database. So at this moment in time, when I looked at this, there were 34,264 assays. So those assays are microarray experiments, so about 34,000. And at the same time, the other database had 414,000, so quite a significant number of samples compared to the Array Express. So this is the GEO, NCBI Gene Expression Omnibus. They adopted Miami as a standard, but then they weren't too keen on the MAJOM standard, because, or the MAJML, the markup language, because they found it was too complicated. So they actually adopted the MAJTab standard. In fact, they were keen for promoting that, and that's the main reason that they managed to get Miami compliant data into this database, and they managed to get the bioscience researchers to actually input the data into GEO in preference to Array Express. So again, it's an example of making it all simpler potentially makes it better, and that over-engineering is possibly not so good. Eventually, GEO and Array Express started to communicate with data, and they actually share data, but the Array Express data is better annotated, and the GEO data has less annotations, but there's more of it. So it's really a balance between the two databases. Okay, so I think that's probably a good point to stop this section, which has been about gene expression data standards. In the next section, we'll move on to a different topic, the BioPax language, which is a formal representation, ontology-based, of biological pathway knowledge. And I hope to show how the two of them sort of link together, and how this is actually, it provides a much more powerful framework for carrying out machine learning on gene expression data. It's not very common at the moment, but it is potentially a much more powerful framework. So this section has really been about gene expression microarray data standards. I hope that I've shown that they involve more than just a floating point array of numbers, that you need to have annotation, metadata about the experiment. MJED Society recognised this in 1999 and actually started to develop a number of standards. The Miami standard, to actually state what is the minimum information required about a microarray experiment, because different researchers were presenting very different things at the time. And then MAJ-OM as the object model based in UML to try and express the complexity of experimental designs and the experimental descriptions of the samples of the genes and how the experiment was carried out. And that's then led automatically from the object model to MAJ-ML, the markup language, which gave a language to transfer this data from one system to another, and then to actually have a way of describing in a compatible manner that everyone understood the terminology being used in the MAJ-ML. They introduced MJED ontology, which is an ontology of gene expression experimental terms. And then there was a bit possibly over-engineered, so it wasn't being used, so they introduced a tabular format. So we've also looked at the array express and the geodata databases, which store gene expression data. Okay, so see you in section 2.